My name is Vladimir Kefalov, and my lab is interested in the physiology of photoreceptor cells. Those are the cells in the retina that detect light and convert it into an electric signal. So they are the cells that drive the first step in the visual process. And of course, as a result, they are very important for our vision, and any deficiency in those cells results in blindness. And so we are studying the function of those cells in normal conditions and also in diseases. So, like I said, photoreceptors are the cells that initiate vision in our eyes, and their function is very important for our normal vision. So we study very basic physiological processes, such as the mechanisms that allow those cells to convert the energy of light into an electric signal, and how mutations in the cascade of reactions that drive this process affect the function of photoreceptors. We also study very basic physiological processes like light adaptation, which is what your eyes do when you go from a dark room to a bright environment, and also dark adaptation, which is what happens with your eyes when you go from bright environment to a dark room. Yeah, we have many projects. We are a very collaborative lab. We have many projects going on at any given time. One project that I'm very excited about is we discovered that a gene, one transcription factor that we can target genetically, makes the rod photoreceptors. Those are the cells that die in diseases such as retinitis pigmentosa. So the targeting of this gene seems to make the rod photoreceptors very resistant to degeneration in several different animal models of this disease. And we are very excited because this points to a potential treatment that will be gene agnostic. In other words, it will allow us to treat patients that have retinitis pigmentosa caused by many different mutations in many different genes. So again, we are very excited about this study, but we have other studies as well. We are largely, for instance, we have a very cool project underway showing that fasting might actually be beneficial for the health of the retina, specifically photoreceptors. So we are exploring this at the moment. We're also looking at the role of autophagy, which is a process where the cells under metabolic stress kind of start digesting their own organelles. And it looks like, again, inducing autophagy by either fasting or exercise might actually be very beneficial for the function of photoreceptor cells. So it's been really amazing several decades of research in our field, which has been driven by the combination of genetic manipulations in animals, mostly mice, and biological studies, in our case, electrophysiological functional studies. And as this has been extremely powerful, and we have learned a lot about what are the mechanisms that drive the function of the cells that we study, the photoreceptor cells, and what are the disease mechanisms when those cells get a mutation and their function goes wrong. One challenge that I think we are coming up to is that mice are nocturnal animals. Their retinas are somewhat similar to the human retina, but also different. They have mostly rod photoreceptors, very few cone photoreceptors. And so one large challenge that I think the field overall is coming into is that there is a limit to what we can learn from studies in mice if we're interested in the function of human retina, specifically cone photoreceptors. And so a couple of things that we're very excited about that we are undergoing projects that we're starting to develop at the moment. One, we are actually switching and starting working with zebrafish. Zebrafish is a beautiful system to study the function of cones because it has about 60% cone photoreceptors. So it's a cone-dominant retina. It has beautiful color vision, and we have some very exciting results from zebrafish showing, for instance, that the function of cone photoreceptors, and those are, again, the cells that we use to see during the day to perceive color and have fine acute spatial resolution. So the function of those cone photoreceptors in zebrafish seems to be highly regulated by the circadian clock. Another project that we are very excited about is actually switching to human studies. So several years ago, we found that the dark adaptation of rod photoreceptors, which is the process that I already introduced, which allows the rods to reset their sensitivity after exposure to bright light, we found that this process in mice is also regulated by the circadian clock, and it's actually more efficient at nighttime compared to daytime. And so we are embarking on a human study trying to measure the dark adaptation of rod photoreceptors in humans, in healthy volunteers, using psychophysical tools in order to test whether human dark adaptation is also modulated by the circadian clock. And this actually has very important implications for people who work at night and who are exposed to bright light at night, because the more efficient this process is, the more susceptible our retinas are to light-induced damage. So people who frequently travel overseas or who frequently work night shifts might actually be more susceptible to light damage